found that while Anthothela grandiflora was really common, scattered in among some of them were these other new species, the genus Latiothela.
Wow, that's really interesting.
Yeah, I think we are.
All right, video.
Yeah, it's amazing when you get the opportunity to do more sampling and you realize that things that look like they are the same might not really be the same. Another reason that collections are...
The value of these Okeanos Explorer dives is, especially when we're coming to new places that have never been explored before, it gives us this first picture. And, you know, we're able to say, based on our past experience, make some pretty good guesses as to the families and the genera and at least what species they're similar to or could be. And this sets us up for coming back in the future if it's a really interesting place and we want to learn more specifically about the diversity. Then we can follow on studies such as the one we're just describing from those canyons, the Norfolk Canyon and Baltimore Canyon. And we do much more intensive sampling and get a much better picture of the diversity, maybe learn something about how the populations that are living here are connected to populations in other areas. And I can't help but remark again, you know, what a fantastic place this is that we are diving on today.
Changing my heading to get
really
diagonal
nice
again
we didn't have any information from this part of the coast.
Sounds good.
And now for the deep sea coral database
so
and
before I
database
think I was heading about
fish database, we've learned a
1,
lot about what are the
330,
potential
so
for species that grow out here. And I've said a few times, and it's been raised in the chat room, that it's remarkable how similar the communities
that heading right there on diagonal, so you get in the middle of the screen, we can start moving along. Although we're going to have to jump pretty
miles
soon.
to the north, and it suggests that there's this broader connection among them, that the currents are such that there may be dispersal of larvae back and forth, at least in the long term, not necessarily the same generation, but over hundreds of years, there is this sort of widespread dispersal and colonization. So really beautiful. Great diversity and pretty good abundance. And as you guys nicely summed up a short time ago, when you get all the factors cooperating, the geology, the amount of food, the currents, and then the right depth zone so that we have the right temperature and other conditions for the particular species that evolved here to survive, then you get these beautiful gardens.
Yeah, it's just amazing. And it also looks similar to those northeast area canyons that have this scarp kind of feature with the ledges and all of the corals that take advantage of the layers and the ledges and living underneath the ledges or straight out like this bamboo coral here. Lots of different microhabitats to find your favorite if you're a coral larvae.
Some of you who are listening along and who aren't party to the many dive planning calls that we have, one of the things that we're often doing is we're looking at the bathymetric maps that the Okeanos Explorer mapping team has made, and we're trying to identify these areas where we might see just this kind of geomorphology, where there's a steep slope, maybe a wall and a ledge, because we know that that kind of underwater feature can accelerate currents moving across it, and this is exactly the sort of thing we want to see. I'm not sure if we looked before. There's some really spindly-looking branch things hanging upside down. They're white.
Yeah, we're seeing some right now. Do you want us to take a look?
I think the pilots are discussing something, so we might need a second here.
Copy that.
Hey, Watchley, this is co-pilot. It's just a shift change right now.
Yeah, we're having a shift change of the pilots, so we're just going to hang out and admire this beautiful scene for a minute or two.
Yeah, what we're seeing actually is the transition from one rock type to another, and you can see how different it makes these overhanging ledges. The overlying rock is a little more massive than those very thin slabs that are breaking off, and even they look like they're tilted down, but that's because they've broken off the flat-lying layer that goes back into the landscape well under these other rocks. So the edges have broken, and then gravity is just causing them to tilt downward.
1923, so a little less than
might make one other attempt if we can, but I don't know how we can take a sample when we're hanging out in the water like this. We tried lower on this scarp to recover a rock, and it disaggregated very rapidly, so we weren't able to get a piece of it. This is a little different from what we tried before. I've seen at least 4 different rocks, not being able to really identify what they are without a closer look. Certainly these rocks on the ledge are somewhat lithified, meaning solid rock, as opposed to they won't crumble when you pick them up. And they're also coated with that iron manganese crust that if you've been on for 3 or 4 days, you've heard that many times. I apologize, but there are so many people around the globe who are tuning in for the first time. That crust is precipitating out of the seawater, and it helps to bind together whatever surface it coats. I just saw a planktonic organism stream by in the flow. So we've just finished our watch change with the front row. We'll reintroduce once they get all settled in. In the meantime, we'll introduce the back row because we have Stephanie here. Stephanie, you want to say hello?
All right, come on in, video.
Hello, this is Stephanie Bush. I work at the Smithsonian Institution National Museum of Natural History, and my background is in water column stuff, which is why I'm not tuning in quite as much on this benthic stuff. But every once in a while I try and guess at a coral.
And Stephanie will be the lead voice you hear on the upcoming midwater transects, which begin as soon as we leave the seafloor on this very dive. So stay tuned and see some graceful and beautiful and very unusual organisms that live in the water column.
And Scott, would this be a very young bamboo coral, or is this a different type of coral?
This is a bamboo coral. It's not young. It's a different species that grows with a very small, thin skeleton.
Wow.
I'm going to guess that this is in the what we call S1 clade. The only species that has been described so far is described by Les Watling from a specimen down in the Bahamas, and it's got the name Cladorisis, and that name refers to how fragile the skeleton is, and it means that it basically Cladorisis, it breaks easily.
Sure looks like it would.
That's what this is. Thank you.
Nice.
Okay, come on out.
That's full wide.
Very nice.
Yeah, let's get the
lithologic change.
Get the other one.
Come on in.
Yep.
Bridge, Nav.
This one looks a little more stout than the last one.
Bridge, Nav.
I'm not sure if that's another one of the Cladorisis that Scott was just
Bridge, I just wanted to confirm our bailout direction is due east.
Yes, we still have the bailout set at due
I'm trying to make the observation.
Copy that, Bridge. You are holding position right now,
This
correct?
has been a hectic
We
dive.
are holding position.
Conditions
are much the same as they have been. It's pretty light. We have the wind and the current
Great,
both on the starboard
pilots.
bow. The forces
Yes,
are
if
about
you could
equal.
indulge me and just zoom in on that intermediate layer that is
Copy that, Bridge. Thank you very
underneath.
much.
If you could zoom in just so that it's in the field of view.
All right, come on in, video.
And focus on that. I'm just...
Sorry, was it the rock?
Maybe
The
a
rock, yeah.
little
Okay.
farther, yeah.
Well, we have a glass sponge there.
Right. Come in tight.
That's great. No, that's fine. I was looking for evidence of flow, which would be indicated by cross-bedding. I don't see any, so there's no point in talking about it, but there's some other organisms that we don't give a lot of attention to because they're small.
That looks like, I'm not sure if that's
Is that a polychaete?
The tube looks so much more
Go ahead.
clear than
So we're
duster
stationary right
type.
now. Are we gonna? Do we want to put another ship move?
Yeah, we can get another move in. Put Sirius over where I am. What have we been moving at?
So we have been moving. The last move was
Thanks,
due
pilot.
north,
That's fine.
0.2 knots at 20 meters. The one before that,
we've been moving along.
350. The one before that was 020.
So this is right on the cliff. So I would say 355, 350 would be good.
Copy that.
That'll move me right over the cliff, though, right? Are we not going along it anymore?
We're
going along it. I'm looking right at it. So, to my starboard is like north. Gotcha. So. I'm turned around. Okay.
So 350.
Yeah, that works.
And we'll go with a 20-meter move.
Sounds great.
Those worm tubes that we just saw on the rock. Oh my gosh.
This is the bridge over.
Never mind. I'm not going to talk about worms right now.
It's 20 meters.
This is great.
Bearing 350.
Okeanos is one of
speed decimal 2 knots.
Fisheries species.
Okay, understand 20 meters, bearing 350, speed decimal 2.
Good copy, bridge.
Let's get mine in video.
Would not be very economical to come all the way out here to get a crab.
Sold that.
This is the golden crab, correct?
Golden
yes.
Crab, yep. Chaseon is the genus. And these are quite large. They're probably at least a foot if you put them down on a...
Yeah, we collected, we chased one with the submersible on the Charleston bump and could not catch up to it.
ROV
and then the next time we saw one, it was mating with its mate, and that was the only reason we were able to collect them. They were very distracted, and we got both of them, and I was shocked when they came up to the surface how large they were. The main carapace
Can I zoom in further?
About a foot in length. This one's a little smaller.
Yeah, I didn't even know that they would occur this far.
Yeah.
Off the coast.
Wandered off track. So he's got some. Are those barnacles on him?
Yeah, it looks like really tiny barnacles.
Yeah, very common.
Oh, you can see the one.
There's one on his back and one on his. I don't know where it's attached, I guess, to the side of his carapace, yeah. Great view of the eye stalks. Are those considered eye stalks, or just they're within part of the carapace?
Wow, beautiful video. I would say this is the biggest crustacean we've seen so far.
There's no question. We had a face-off yesterday with a hermit crab.
Yes.
That movie is coming to a YouTube near you in the next few days. The video team has put some wonderful short videos of some of the highlight features of our videos, one for each dive. So be sure to keep up with the website and look at the video and image gallery, and you'll start seeing that being populated if it hasn't been already. Since it's the weekend, I don't know how much the onshore web team is working towards that, but
Yeah,
certainly this week there will be some tremendous and very entertaining video and plenty of incredible still images from these dives we've had, particularly this one.
Yeah, and so thinking back, before the previous leg of Okeanos, this area wasn't even mapped well.
Right.
And so we've come such a long way towards figuring out that this is really good, important coral habitat and sponge habitat. And I mean, just in a couple of months, we've learned so much. And I guess it's now a lot less of a mystery. I mean, when we were planning these dives a few months ago, you know, it was all wide open. We didn't even know where there might be really good habitat.
Thank you, Bridge.
Whether it was quite as suitable as it obviously is. So it is absolutely amazing to see how much we can learn with just couple of passes with the multi-beam sonar mapping and then eyes on the habitat.
And we should emphasize what a community-wide effort this has been. The call for proposed dive sites was put out in February with a deadline of April, and well over 100 dive sites were proposed by the scientific community. And with each proposal had to be rationale, of course, and what was expected of the dive, what was anticipated. And in most cases, the strongest rationale was we simply don't know. It's truly unexplored. So that is the primary mission of the Okeanos Explorer, and we certainly are finding some unexplored territory and learning a lot, as Cheryl said. In fact, one of the sites proposed, if a site is proposed that we don't have high quality, high resolution mapping, this is a crinoid we're looking at.
Crinoid on I would imagine that might be a Paramaricia coral and a squat lobster.
Squat lobster, yeah. But if we don't have high resolution maps, then the ship visited some of the sites that we thought we would dive to, and on the last leg of of this overall
maybe
mission,
a little more north.
They did detailed mapping of some of the proposed sites, and one of the sites turned out to be completely flat.
So it's right
even though
here,
the Google Earth image did not show that. We thought there would be some sort of topography down there. It was completely flat. So
approximately
we have to be careful. You can't rely on the older maps, clearly.
And you're ready.
I won't go into all the mapping details, but the high-resolution maps are so much better.
Yeah, because I know they're
they also provide some information on the character of the seafloor. If we think there's hard rock or soft sediment substrates, or sorry, go ahead, Scott.
Yeah, sorry to interrupt. Before we leave, can we get a look on the top of this ledge? We were just, I don't know now.
Up on the flats?
I guess. Maybe we went too far.
What was it that you saw, Scott?
There was a really scrawny branched bamboo coral that looked almost exactly like
Co-pilot. Looks like there's that was just
I think
another one of the
It
waypoints. It looks like they wanted to go all the way to waypoint 4.
We actually have a waypoint 5
too. It's so interesting that it was described in the Bahamas
all the way over here.
So
but we're definitely not going to make it
here. And almost
I didn't know what their plan was.
Well, I'll ask them if they want to cut over or cut west now or later.
The consistency with which you might find these things in a particular slice of the ocean.
So is this it here, Scott?
Yeah, exactly. That wispy thing in the center there.
Would that be one that would be a good collection, or?
No, we don't need to collect it. I just wanted to get an observation of it.
Okay, great.
Thank you.
Really interesting.
Watch leads, Nav. I'm not sure if you can see our high pack right now, but
before, Leslie, you were asking about what's the
waypoints
periodicity with which you go from node to internode. How many nodes
not
are
sure
there?
if you want to keep heading
and
north
on this
or
one, I can't answer
cut
that
west
question.
now.
But what's interesting here and what makes it so easily identifiable once you get an eye for it is how far it goes before it branches. You see there are really big intervals between where there's one branch and another. The which always makes it a challenge when you want an ROV pilot to collect it. You want a touch point. It ends up being these very long,
well, actually, pieces. I'm not sure if you're replying, but I can't hear you. Oh, okay. Copy that.
Thanks, Scott.
Sure.
Oh, there's one of the...
Will do. Yep.
Is that a tube anemone there, or...?
Yeah, ready whenever you guys are.
No, that would be a cup coral.
That's the cup coral. Okay,
so another, what, 355 pilot?
Focus. I
I'd
can't tell them apart yet.
Due north.
Oh, and there's a nice brittle star.
Move him off the wall a little bit.
That might be a Giovannia cup coral. I think that's different than the ones we were seeing.
Another 20 meter move?
Sure. Sounds good.
Underneath
right,
those
I'll call it in.
ledges.
Mm-hmm. Yeah, let's come on out.
Bridge, Nav.
Bridge, over.
I'd like to request another ship move. Distance 20 meters, bearing
our last shot of
000 degrees.
The golden crab.
Speed decimal 2 knots.
Chaseon?
Understood.
Correct.
20 meters, due north 000.
Hopefully we'll see a few more.
2.
Good copy, Bridge.
Wow, it's just so interesting to have found all of these corals here and learned so much about the potential for corals in this whole area.
Incredible.
I mean, this escarpment goes a very long ways.
A very long ways, yes.
So it could...
And roughly at this depth,
initiated.
it's very flat on the plateau, so that the edge of the plateau is between 1,200 and maybe 1,500 meters. At different points, it's probably the steepest areas are within that depth range. And then farther south, it descends almost vertically from to a depth of 5,000 meters.
Wow.
And we're not going there. Of course, I'd love to.
Can we kill lasers?
I know that others have been there. I think there were some Alvin dives.
Thank you.
Down in that area.
So I'm not trying to make a shameless plug, but I'm going to tell you that the observations that we've made in this dive today have me really excited to see what we're going to see in the canyons in the next week and a half as we go further north, because I've made these observations a couple of times that there's a lot of similarities to what we see here with the offshore northern seamounts, but not necessarily with all the canyons we see. So it'll be really interesting to see as we transition north, do we start to see a change in the species, the diversity, or are we going to get to see some of these things again? So very exciting.
So true, Scott.
I suggest everyone tune in and find out with us.
Yep, everybody, come on back.
Hey, there's a wire coral that just went off the lower part of the screen. If we could have a look at that. I don't think it's a bamboo coral. Excuse me, I don't think it's a black coral. So I'm wondering if it might be a primnoid whip.
And which type was it?
It's an unbranched kind of a...
Yeah, I think we found it.
Coral. It's white, just coming into view now,
yes.
Just below the Lophelia or Solenismilia.
Solenismilia, yeah.
Yeah, that thing. I'm not sure if that's a bamboo coral or a primnoid coral.
Oh.
Come on,
or maybe it's a black coral.
I didn't even know there was a primnoid that had a pattern like this.
Definitely not the yellow or the orange black coral we've been seeing today.
Right. Doesn't look like a black coral.
Bamboo.
Yeah,
oh,
definitely
bamboo,
a bamboo.
Yeah. You can see the bamboo,
coral.
yeah.
Yeah, it's a bamboo coral. Great, so another different one. This one is an unbranched one. Great diversity today.
Yeah, that's great.
We'll have to sum up the numbers of species for each of these groups, just so...
Yeah,
I'm sure, of course, the biologists will be doing that.
This one has some eggs at the base of the polyp. Every once in a while you see a polyp and it's got some bright white right at the base against the skeleton. Those were probably gonadal material.
Wow, and that's so unusual to be able to catch that. We know so little about when deep-sea corals reproduce.
Can we pan to the right, please, to the base of this coral?
Absolutely.
Thanks.
Yeah, I can see some there.
ROV nav bridge move complete.
Thank you, Bridge.
Another cup coral in the background.
Yeah.
Definitely bamboo coral.
Great, thanks.
Right, let's come at it.
And the pink corals that you're seeing hanging out, those are the stony corals, the scleractinians that we're seeing throughout this area.
Go ahead. Yes. Do we have the ship moving?
The ship just stopped, I believe.
All right.
Okay, we
Just
are
stopped.
looking
Let's
at a
bring up hydraulics.
very huge, beautiful Liopathes black coral here.
Ramping up hydraulics.
And this is a species that we're
And
including
then we're going to go in one of the starboard bio boxes.
So we're going to try to take a small sample of this. And Liopathes is quite common in the Gulf of Mexico at certain depths. I don't know, I've seen it at shallower depths than this, but I think it has quite a vast distribution.
Yeah, it's interesting. I think my student, at the time, my student Mercer Brugler did an analysis of the video we took for the New England seamounts, and my recollection was, it was 1,500. I was going to say Liopathes was always shallower than 500 meters, but no, it was always shallower than 1,500 meters.
All right, you're here at 90. We should have plenty of time, though, if the ship stopped.
All right, excellent.
Hydraulics are ramped up, and we're ready for the door whenever you are.
Let's do some zooms here. Get some shots while we have this set up.
Beautiful. So we're at a depth of 1248 meters right now. We're on the edge of the Blake Plateau, so that's pretty much its average depth at the edge. And we are at its edge called the Blake Escarpment. And if you tuned in on our first dive, which was now 3 days ago, we were north of here, considerably north of here, a little greater depth,
so just
1750 meters.
Copy that.
And saw very different topography, much lower slope with only a few outcropping rocks. But as expected, wherever we find rocks, we find diverse and rather abundant biota. Nothing at all in comparison to what we've seen today, though. The diversity is incredible.
That's full wide.
So that's full wide. I can back up if you want if you want to see it top to bottom. I'm a little close to it right now.
I think that's okay. We can definitely get a sense of the scale of this.
Do some pans.
Brittle star over on the left.
I just want to let you know, if we are going to try to collect this, it really needs to be cut and not try to be pulled or bent. It's got a very tough skeleton. And I'll make the note the comments are coming up in the chat room about the age of Liopathes, and so there's been a variety of groups who are interested in aging deep-sea corals for different reasons. And so far, the oldest known coral was a Liopathes black coral that was sampled in Hawaii. And I don't have the paper right in front of me, but I recall that the age was something like 4,200 years old.
Yep, that's what I remember too, Scott.
And still living.
Yep.
The good thing is that they're asexual, so they can continue to grow after.
It's incredible.
So, can we bring up Mini Zeus on your screen?
Do you want me to take control?
No, bring it up on your screen. Yeah. Yes. Yeah.
Co-pilot, I'll monitor your Zeus.
Copy that, thank you.
Yeah, this species gets quite large. I've seen them that were about as tall as me.
So I'm just going to take the top of this, if that's okay with you, Science.
And Les Watling has
that's okay. We
mentioned
don't need
that
much,
Liopathes off Hawaii aged at 4,900 years, and probably most of them are in the 2,000-year category. We're getting requests to measure the base. So after we do our collection, we'll try to get in there and do that.
Co-pilot, the starboard bio boxers are the only ones free.
Copy that.
So you'll want to extend the pin.
Already done. Ready to extend the door?
Yep, we're clear.
Coming out.
Just make sure we don't hit the coral here.
Copy that.
This is also the species I was referring to earlier that produces copious amounts of mucus, so you may enjoy pulling this out of the biobox later.
I'll leave that to the biology team.
Oh, goody.
I've got rocks to take care of.
Someone, Scott will make sure we have someone with a camera to document the amount of, the copious amount of mucus.
I want to have the camera be focused on your faces instead.
Yeah, that's good. Excellent.
Stuff in there?
Yep.
Okay. Closing the drawer?
Yep. All right, that went in starboard outer.
Great, thank you. That was a beautiful collection.
Drawer is closed. Returning Menzies to the middle.
Yep. All right, I'm going to get a shot of the base now.
Great, that would be wonderful.
Okay. Would you mind if I change the view back to
go ahead.
Sirius? Switching back to Sirius.
Watch lead nav.
Excellent collection, pilot.
Thank you.
Just want to confirm that's a specimen 09 bio, correct?
Right, let's come in.
Thank you.
We want to get the lasers in the
Oh,
base.
sorry, I thought they just wanted the base.
Yeah.
Some gastropod shells, just in a little pit behind.
Let's get a specimen shot here.
Would you like lasers off?
No, you can leave them on.
So the diameter of the base of that skeleton was really quite impressive. I would say at least 8 centimeters.
I might go past the skids.
Yeah,
and the base of the skeleton itself was just above the holdfast. I mean, the main part of the axis going up, kind of like the trunk, if you will, was quite thick, which also speaks to how old this is. You can see at the tips of these branches, at the tips of the colony, the branches are really, really thin, and they grow like a tree, but they continually deposit new layers. We don't know what the periodicity is exactly of those layers. Like, I can't say that they're annual layers, but they are periodically laying down a new thickness of skeleton. And so it takes a lot of time.
We get the waypoint on that?
Yeah. Which just speaks again to how old it is.
Watch leads nav. So we have about 27 more minutes of bottom time. Copy that. So star heading to waypoint 5. Copy that. We'll do that.
And if you want to turn your heading towards waypoint 5.
Copy that.
And then I'll center up on you.
Oh, wow. Is that a little basket star?
It looks like a basket star.
So
before we head in,
now,
pilot, could we pause and do a quick zoom
here?
Yes,
330.
All right,
Absolutely.
I'm moving from current position to 330. You may go out of view, pilot.
That's fine. All right,
On
let's
at least.
zoom here, and then we'll get the lower one.
Basket star.
Maybe 2 basket stars.
That's beautiful.
Okay, maybe we should do those. Yeah, thank you.
That is beautiful.
Should we do a Niskin bottle collection
and
as well?
this is what, the second, maybe the third of these candelabra bamboo corals we've seen today?
Go ahead.
Certainly the second, if not the third.
Copy that. So if you want to work with nav, co-pilot, we can do that.
I'm ready when you are, Nav.
So these basket
Copy that.
stars pretty much lay on their backs on the sediment or some other area and
looks like
have their tentacles facing up
4
to
and
grasp
5 left.
particles
The other one come out just a little
moving
bit.
by.
So I don't see
This
any
one's
local high
behaving a little
on
differently
D2.
from what I've seen
D2 is at
do in the
266. That seems
And
like it's
here's a
going
whole
to bring us
tangle of them.
right
Oh my
over
gosh.
the,
Oh my gosh.
pretty much due west. So I'd say we just go that direction.
Yeah, in case
least for the
some
first move.
viewers that
At
don't realize,
266.
these are
Yeah,
brittle stars,
D2's coming
opioid
right in.
brittle stars, so actually
Copy that. I
closely
will move
related
back
to some of the ones
266.
that we've been seeing
The
with their arms
science also asked that we fire the last 2 Niskin
on
bottles.
the corals, like on
Are
the
you prepared to take those?
It's just that they have their arms continually bifurcate as they grow, but if you saw them very young, it would
Lasers
look
coming off.
much like a
Go ahead,
regular
survey.
opioid brittle star.
We have
It's
not
also worth
yet.
noting
We'll let
that
you
they
know.
are suspension feeders, so they will unfurl the arms as you see
Okay,
here.
co-pilot,
and
I'd say
extend
let's
them
go
into the
ahead
water
and do
column
one.
Specifically are grabbing their prey
Resetting the
from
Niskin
water
bottle
column.
number 4. And firing Niskin bottle 4 now.
Do you have hydraulics
Survey.
on?
We just
Niskin
fired
bottle
Niskin
4.
bottle 4.
Okay, good.
Would we like a 5th or save it?
Stand by, co-pilot. I'm going to record that sample.
Standing by.
Great close up. So are there 2 in this little group or 3?
Yeah, let's come out and get the
I
specimen.
can't tell
I thought there was a second one
in that tangle
in
of
here.
arms.
It can't all be one.
Just keeps going.
Maybe it is. Huh.
I think there may be at least 2. It looks like there may be one below the.
Yeah, I thought I saw a second.
So this is the second one.
width of the thickest arms.
So
Yeah, there's definitely 2 on the whole coral.
Yeah, no, I meant.
But
Okay,
I
co-pilot,
thought
you
there
go
were
ahead and fire
2
that other one.
bundled.
On top of
Okay.
the other,
Yeah.
Resetting the 5th
a little bit
Niskin
farther
bottle.
away.
Yeah.
So
And
in
firing?
Glacier Bay, it looked like some
Number
of
5
these
is
basket
fired.
stars were actually eating the
1907
coral
UTC.
polyps.
Copy that, co-pilot.
It does look like there's some coral
Survey 1,
skeleton that's
Nav.
exposed. Any idea whether that's a common
So
thing
that
around
was the
here
last
as well?
Niskin
Or?
bottle. So that would be D2, dive 04, specimen
I'm
11,
sorry, could you repeat the
W18.
question, Cheryl?
Are the basket stars
Thank
like
you.
this known to predate on the corals, eat their polyps?
Hydraulics
Does it
have
look
been
as if there's some
good copy.
exposed skeleton above, and we were kind of thinking that was what was happening in Glacier Bay.
I see. As far as I know, all of the shallower water basket stars are suspension feeders. They're feeding out of the water column. That one that we see on the upper side screen is a good example. Extends the arms out and fishes from the water column. So I would think that the damage that we're seeing to the colony is simply abrasion because the basket star is sort of moving back and forth against the polyps and so sort of scraping them off. But as far as I know, there's no evidence that they actually feed on what they're sitting on.
Okay, and it could be a different species.
Okay, pilot,
there
so
as
I'm going to
well.
put in a move. What do you say we start with a 30-meter move
Okay, should we move on, you think?
at
Yes.
due
Okay.
west?
Pilot, thank you.
Sounds good.
I'd be comfortable with that.
Come what? And we can lose the skids.
I am lined up at 265 at the moment, pilot.
Copy.
Bridge, Nav.
This is the bridge, over.
I'd like to request a ship move. Distance 30 meters. Bearing 270 degrees. Speed .2 knots.
Okay,
As we are
will
nearing
move
the end of our
30
dive, we're going
meters
to
at a bearing of
traverse the
270
flat
degrees, speed
top of this
Good copy,
scarp
Bridge.
feature that we've been very slowly and carefully ascending for the past many hours. We're still seeing plenty of diversity. So it's certainly still good habitat, some prime habitat. There's another of the candelabra.
Candelabra. It looks like a
Bamboo
Liopathes
coral.
ROV nav, bridge, move initiated.
So this is still very good habitat.
Thank you, bridge.
The rock is very different here, at least its character. It's very rugged and almost swirling areas, as though currents had somehow, I don't know, eroded pits within the surfaces.
And as we're looking at this beautiful scenery, I wanted to say Happy Father's Day to all the fathers out there, especially mine, Steve Morrison, if he's listening. And also, happy birthday to our expedition coordinator. It is her birthday today, so everybody give her a shout out.
And she is wearing quite a hat. It's a birthday cake hat that is on board the vessel for the lucky few who have their birthdays while we're out at sea.
And hopefully her birthday present of this beautiful coral area that was a mystery to us just a little while ago will be just what she wanted.
That's right.
I have you there at 45. I can come down a little bit if you want some more, but
we did say her name right. Casey Cantwell. We
Casey
just said
Cantwell.
her. Okay.
Oh, that's right.
Yeah,
I don't know if we actually said her name. Casey
move.
Cantwell. She's a fabulous lead for this whole expedition. Been incredible assistance to the three of us, to Cheryl and Stephanie and me, because we have had such incredibly steep learning curves to get ready for all these dives and to get used to all the controls and the technology. But incredible support team everywhere around us. The ROV team is exceptionally nice to work with and helpful, and the videography team is, as you will see in the upcoming videos, they are something special. And I've never worked with quite such professional and friendly teams as these, ROV and the video teams.
And the amount of work that Casey does leading up to one of these expeditions is just absolutely amazing. All of the different moving parts have to come together at the right time, and so much information has to be digested from the collaborators who suggest dives and things like that, and to all of us who are going to be out here relaying so many communications.
Zoom on that, please, Pilot?
Would you like a zoom on?
Right in the center, just a quick one, and then we can maybe look over to that branching sponge.
All right, come on in,
as well.
Is it the sponge or the pink coral?
I'm not sure. Do you want to see the sponge watch lead? Or?
Just for, I was talking about the one farther up that was in the center of the screen. That's fine. Yeah, that rock with several different organisms.
I wonder if that's the Faria coral, a sponge that we've seen. But this one looks a little bit more round instead of long.
So we're getting close to the end of that move, pilots.
Come on in video.
Oh no, I'm sorry, we have another 10 meters
It's
to go.
another bamboo coral. Looks like a Selenosmilia behind the sponge. The sponge has, is that sediment that's sitting on it? Ooh, and there's another squat lobster within the sponge maybe. Or is that a...
I think it's a brittle star.
Brittle star.
Yeah, on the right. There are several of them, actually. I see a few other arms sticking out of the different holes within the sponge.
Bamboo coral.
I'm coming down just a
squat
tad
lobster.
there, pilot, so you can go over and see that branch sponge. We have a delta of 16.
I always think of that song, Rock Lobster. I don't know.
Pretty benign out
Yeah,
right now.
you remember that?
Yes, I sure do.
We need a new one called Squat Lobster.
So, Tina is asking about a dark red coral to the left. I'm not sure if that's in this view or...
It's
So
come
maybe
out partial.
Yeah. We might have passed it, but...
Pilots and watch leads, be advised, we have about 10 more minutes on the bottom.
Yeah.
Roger
Copy
that.
now.
We only have about 10 more minutes on the bottom, so we will take quick glance as far as we
ROV nav
can
bridge move complete.
across this flat
Thank you,
plateau
Bridge.
at the top of the escarpment here.
So I think we can fit in one more move, pilots. You want to go ahead?
Yeah, you can go ahead and put that in.
All right, another 30 meters at 270.
Sounds good.
Copy
Come
that.
wide.
Bridge, nav.
Bridge over.
Like to request a ship move. Distance 30 meters. Bearing 270 degrees. Speed decimal 2 knots.
Understood. 30 meters, 270 degrees, speed decimal 2 over.
Good copy, Bridge.
Wow, that's an interesting sponge.
I think the one we just crossed is similar too, but this
Yeah.
one is a little more
I see that Chris Kelly just
more
joined
full.
the chat
Oh,
room.
hello, Chris.
ROV nav
Maybe
bridge
he
move
could
initiated.
inform us about this.
Come on,
Thank you,
NVIDIA.
Bridge.
Wow,
That is, yeah, we have not seen that unless
that's
it's
crazy.
grown differently. You know, that almost looks like the dead one that was sticking out that we had the
Zoanthids.
zoanthids, yes. Oh, and look, it has zoanthids on it.
Yeah, Chris wanted to collect this, but unfortunately we're out of bio boxes.
Did we fill all the bio boxes?
I believe we have a starboard inner.
Starboard inner open?
Yes, it is.
Yeah, we have one bio box. Watch lead.
Oh, there is one bio box. Would there be time to...
Yes.
Okay, well,
Yeah,
Chris,
how much time do we have?
we might
We
be able
have
to collect...
8 minutes,
Oh, it's plenty of time. All right.
counting setup. So,
Yeah,
a
come
little
on
tight.
out. Let's get lasers on. And then get
There's
hydraulics
another
going.
one of
Lasers
them
coming
that's
on.
dead there.
Mm-hmm.
All right, I'm
Yeah,
going to stop
I think
the
it
ship,
is the same
pilot.
thing.
Yeah, go ahead.
They're very similar.
So do we want to sample this?
Bridge, nav.
Yes.
Okay.
Bridge over.
Would you come to an
I
easy
noticed the
stop,
live
please?
one also had the
Yes.
zoanthids or anemones
Bridge.
or whatever they are on them, so that would be a valuable collection.
Right.
All right, come back in on Zoom. So they want something with the zoanthids.
ROV nav bridge, all stopped.
Thank you, Bridge.
Yeah, I don't see a way. Yeah. All right, let's come out.
Hydraulics are ramped up.
Yeah,
Yours
good copy.
ready to extend. We're going to starboard inner.
Good copy.
Hi guys, this is Chris.
Hi, Chris. How are you?
I'm doing good. I'm sorry I'm late.
Oh, it's okay.
Bringing up Mini Zeus on my screen.
Came in at a good time, at least.
Yeah, this sponge, so Nolan
And taking control
was
of
on,
Mini Zeus.
I guess, previous cruise. He's been sending me pictures of some of these Atlantic sponges, are the ones that were in the Gulf. And he sent me a picture of this thing, and I really have absolutely no idea what it is. I can't even go to family. Scott suggested Korea, and that is a possibility. But it could also be a couple other things.
All right, drawers
I've
clear.
never seen
Doors
anything
extending?
like this before, and I was just astonished when you just came up on it.
Oh no.
Wow, that's awesome. I hadn't seen anything like it either, but I don't have your experience, so. Ooh.
Yeah, this is at least unusual to me. I've never
Doors
seen
fully
anything
extended?
like this in the Pacific, and I haven't seen anything like this
Following
in my
the Mini
reference
Zeus?
It's
material.
very delicate.
So this
Yeah,
could
tilt
be
down,
something
tilt
brand new for all I know.
Wow, that's great. Uh-oh.
Yeah, thanks so much for the last minute request here. I'm certainly going to try to participate at least an hour or two early in the morning before work from now on, because it's beautiful. Really, I wish they would have seen today, because I have been on for a little while here and saw just some beautiful corals and other things that you guys came across.
Closing the box.
It's been an incredible day, Chris. And we look forward to you joining us whenever you can.
All right, that's in the box.
Yeah, I'll definitely try. So thanks again, guys. I really
That was a
Sure.
quick reaction.
Yeah, and Sandra Book pointed out that we
Go
didn't
ahead.
see anything like this in the mid-Atlantic canyons, even though Les Watling said that it's actually somewhat common in the New England seamounts.
How much time do we have, Andy?
wonder if Les knows whether it's ever been collected before.
We have to get set up.
Okay. Fortunately, we're out of time, Watchley.
I don't think that we made any
Right,
collections
you can bring
of
your
the
camera
sponges
back.
while we did the work on the New England seamounts, Chris.
Okay, so this is a very important
Yeah, let's
sample
stow
then.
the swing arms.
Copy that.
Yeah, that's just really exciting. I definitely think that has potential to be a new species. And again, I really don't know. There's probably at least 3 different families that it could be in.
Wow.
And it's great that one definitely had zoanthids in it on the part that I believe was collected, so that'll be a valuable addition.
That's wonderful.
Terrific. Yeah, we've seen more and more as we've come across and been able to dive on some of these deeper areas. We've seen more and more sponges that have these zoanthids in, or other types of anemones. There's one type of anemone that was identified from Nadia Samarian, I think, in Moscow. As I don't think she's in Moscow; she's in Russia. But anyway, Spongiactis, I think, is what she called it. It's actually a commensal for certain types of sponges, and we've seen these on a bunch of things. The Farid sponges, like Scott suggested, and a number of other things. So there's something going on between these anemones and zoanthids and these glass sponges.
Huh.
Let's zoom in on this sea star while we're stowing the swing arms.
Yep, we're getting ready to leave the bottom, unfortunately. It's been an amazing day today. We have really learned so much about the biota of this area and the Blake Escarpment
Bridge, be advised, the ROV is coming off the bottom to start ascending for
it looks great.
the transects.
And I'm going to try to get the YouTube video of this.
Say again your last?
Take a look at that later
ROV is
today.
off bottom. And we're going to ascend and then begin transects.
Chris, there's so
At
much in this
1,300
video
meters.
that we've collected
sorry,
today
at,
that it's going to take a long
950
time
meters.
for biologists to go
Roger
through
that,
it
thank
and
you.
identify everything we saw. The corals, the sponges, and all the associates. This is just an amazing place for diversity. I don't know if I've ever had been on a dive with this much coral
Coming out
diversity in one dive.
Swing arms are stowed.
It's rather astounding.
Yeah, and I think you're going to need to
Wow,
turn
very cool.
to your...
Okay, well, I get up really early in the
port, I
morning. I'm
believe?
usually up around 4:30,
Ramping
so it's going to get turned on every morning here.
Okay,
Yes.
great. We'll look forward to it, Chris.
I can
Yeah,
turn to
thanks,
port.
guys.
So you
Thanks
can go
for
ahead and do that.
tuning in, Chris.
Tomorrow.
Spin into port.